This is a presentation on selective breeding, genetic engineering, and cloning. Selective breeding is when people breed for a specific trait. This can be done with plants or animals. It is not part of natural selection or evolution. It can lead to certain health problems in an organism, and I'll give you a couple of examples coming up. But one of the important things to note is what would happen to those animals if people disappeared, or those plants if people disappeared. Here's an example of an organism that has been selectively bred. This is a German Shepherd. German Shepherds are known to have hip dysplasia. Basically what has happened is their body has become too big for their hips to support, and a lot of times when they get older, they have health problems with their hips. So why did we do it, and what are the benefits? Usually when people selectively breed, they're doing it because they want specific traits in the organism. So the benefits are those specific traits. Whatever people believe are good traits in an organism, those are the traits that they breed for, and they believe that that is beneficial. Here's an example of selective breeding in strawberries. Here you can see wild strawberries. Wild strawberries are actually quite small, but if you notice when you go to the supermarket and you buy strawberries, they're much, much bigger. What has happened over time through selective breeding is that strawberries have been selectively bred to be bigger. We've also selectively bred tomatoes. We've also genetically modified tomatoes, which I will talk about that in a moment. We also selectively breed livestock so that they have the characteristics that we would like. For pigs, they may be leaner so that they produce more meat. For cows, it may be that they produce more milk, just to give a couple of examples. So we as humans have made new varieties or breeds that may not have been made through natural selection. How we do that is by simply choosing traits and breeding two organisms that have those traits to get offspring with those specific traits. Genetic engineering is the deliberate alteration of the genetic material of an organism. So now we're talking about people changing the genes of organisms. What they will do is insert copies of genes from one organism into another. And that's called genetic recombination. The genes have been recombined using technology. Sometimes we've even taken human genes and inserted them into other organisms. A perfect example of this is we have taken the human gene for making insulin and inserted it into bacteria. So now there's bacteria that can make the insulin, and this is used in medicine for people with diabetes. We can also do this with foods. A lot of recent genetic engineering has been in crops, and what scientists will do is take a gene or create a gene. For example, a gene for an insecticide will be inserted into the crop. So instead of spraying insecticides or needing to spray insecticides onto the crop, it's already in the plant. This could become a major problem because remember that crops make seeds, and those seeds could blow around, and eventually these seeds could be out in nature, out in the environment, and we don't know some of the consequences of that. We also don't know some of the consequences of eating genetically modified organisms, also called GMOs. There's been a lot of controversy, a lot of scientific research going into this right now, but I can guarantee you that most of the food that you eat is genetically modified. It is not a requirement by the government that these foods be labeled as genetically modified. We're going to talk about restriction enzymes a little bit more in more detail in class, but what a restriction enzyme does is that it will cut DNA at a specific location, and then this allows the scientist to insert a specific gene into the DNA of an organism. So that's just how we're able to do genetic modification and genetic engineering. So here's an example of corn. I used the example of insecticides already, pesticides, and again, we'll go into a little bit more detail in class. This is not a real cow, but if you look at this cow, it shows a map of the world. It's just an example of the degree of control that humans are starting to have over organisms. Okay, one day we may decide, or we may be able to control every single phenotype about an organism, including what color their hair is and what the location of that hair is. So it's just an example of what may come in the future. There are some ethical issues with genetic engineering. Their safety. Okay, genetic engineering has only been around since the late 1990s. Genetically modified foods, crops, organisms. So we're not real sure about the long-term effects of genetically modified organisms, genetic engineering. Another ethical issue is, what are the human rights? Again, I told you that a lot of these foods that we eat from the supermarket aren't labeled as genetically modified, and yet they are genetically modified. Is it a right of ours to know that they are genetically modified and decide for ourselves if this is something we want to be eating and putting into our bodies? Another issue is property laws. A lot of companies are claiming the rights or patenting specific genes. Therefore, who's to say that they own it? Is it a natural gene? There's a lot of ethical issues going into genetic engineering. But a positive of genetic engineering is gene therapy. And this is when scientists will insert a normal gene in place of a mutated gene. And what they will do is they will use viruses. And I've explained this example before, but I'll say it again. A perfect example for cystic fibrosis, they will inject the missing gene into the cold virus, and then that cold virus will go and insert the missing gene into the cells, and then those cells can start making the protein that is missing. Some of the drawbacks is that, it's not necessarily a cure. It costs a lot of money. And right now, a lot more research is needed. Lastly, I want to talk about cloning. And this is when an organism that is genetically identical to another that is pre-existing is made. The first clone was Dolly in 1997. Asexual reproduction is a form of cloning, but when we talk about cloning in biotechnology, we're talking about scientists doing cloning. But bacteria do asexual reproduction, okay? An organism that is genetically identical to the parent is made. Budding organisms also do asexual reproduction, and that is a form of cloning. The problem with clones made by scientists is that they do not survive very long, and there are usually problems with normal growth and development. And this usually occurs because of the cell that the scientists use in cloning is older, and therefore the genetic information is older, and there's some science behind why it just doesn't quite work. One of the reasons that it may not quite work is because of genomic imprinting. Different genes are active in different cells, and they're also active at different times. So they believe that one of the reasons that clones aren't as healthy and don't live as long as the original organism is because of genomic imprinting. the genes aren't functioning when they're supposed to, and or the genes in the specific cell that they use are already too specific to that cell. So I just want to go through this image of cloning and how they cloned Dolly, the sheep. So what they did was they took a cell from a sheep's udder, and then they took an egg cell from another sheep. And what they did is they removed the nucleus of the egg cell, and they inserted the nucleus of the donor cell. So what ended up happening was this newly formed egg cell had the exact same genetic makeup as the donor. They then basically gave the egg cell an electric shock to get it to begin mitosis, and it started forming it into an embryo. That embryo was inserted into a surrogate sheep, and eventually that surrogate sheep gave birth, and we ended up with the cloned animal. This animal here is genetically identical to the donor. Please complete these 3 questions on a loose leaf piece of paper.